I am Valderiz Dutra from the Federal University of Minas Gerais in Brazil, and I have a question for Elena and one for Thomas. Elena, my question for you is, you show that different seaweeds have action in the worm, but I was wondering if they have the same mode of action. If they interfere with the same part of the life cycle. That was one question for you. And Thomas, I noted that you used dexamethasone to improve the clearance of your drug, and I was wondering if that has the better result that you found, if it has anything to do with the fact that dexamethasone may interfere with the immune response, and therefore it could shift your response towards the Th2, which is protective in schisto.
Okay. Well, about the mode of action of seaweeds, first we started with extracts, and it's a new study, 4 years, to screen more than 40 species, and we had now some chemical frameworks to choose to follow with the studies. So we don't know the mode of action for schistosoma, for worms. We started with tests with adult worms. We still didn't test it in schistosoma. And interesting, we have activity of some compounds both in worms and in the snails. But in this first stage, we are trying to identify the chemical structure. We are synthesizing analogs to do studies between the relationships and the structure, chemical structure, and activity. So we can know a bit more about the mode of action. Thank you.
Okay. So we haven't looked too much at the immune system aspects in the S-manzoni mouse model, but we had tested the dexamethasone on the worm itself, so we know that there is no interaction at that level. That's the only answer I can actually give you on the immune system. Does that help? So well, we can. Yeah, sure.
Any more questions?
I have.
Yeah.
Yeah, go for it.
So, can we get those compounds?
We have the amount we isolate from the crude extract is a small quantity, but we are trying to synthesize in laboratory, but it's hard, it's difficult, because nature is not friendly with chemists. So it's not easy to obtain these molecules in the laboratory, but we are trying to synthesize. But we can.
But the extract, how much you get?
Less than 0.7%, I think, from the pure compound from the crude extract.
Like in micromolar?
We can obtain amount enough to do all of tests, cytotoxicity and so on. I think it's not a problem. And I have a question for you. Is it too difficult to obtain a crystal from protein plus a compound?
If the protein the crystal likes to be soaked, not a problem. If it doesn't, yeah. So, but in principle, if you have a crystal, in principle, you could try it out. But you have to have the protein.
Yes, we
Okay.
okay, thank you.
Any more questions from the audience?
Hello, Beatrice from the Merck Global Health Institute, working with Thomas. Actually, I have a question for the three of you. What, in your opinion, is going to be the next challenge you are going to be facing, moving your compounds through preclinical discovery, preclinical, and how are you going to translate that into the human situation?
So the animals first. So will give us some ideas on how compounds will show some activity. I think it's really building on the knowledge with praziquantel that will probably help most. One of the challenges I can see, for example, especially if you target the juvenile worms, is how to read the PD outcome. I think that could be maybe one of the I would say the risks in developing that strategy.
Yeah, I have no comment. I'm 15 years before that. I would love to see those models, like non-animal models, available, because then you could do really exhaustive searches as opposed to having to worry too much and worrying off.
So just building on your work on the TGR, if I remember correctly, there is a group in North America that did do some work. I think it's Williams group,
Yes.
right?
Yeah.
How does it translate? I mean, it did not really translate into much efficacy, right?
I don't think so. They're in the early discovery stages.
Stages, still.
Yeah. But they do have phenotypic assays, so they do have possibly other work.
I'd like to ask a question. From a partnership perspective, you're here from São Paulo. You have novel compounds outside of praziquantel, outside of niclosamide. Are you looking for partners? Do you have any kind of message for them out here? It seems as if it should be something like a consortium should be built around what you're doing potentially. Do you have anything to say to them out there? Are you looking for partners? And if so, what would be the profile?
Actually, well, as a non-Brazilian, I can maybe answer. There is quite a large meeting beginning August, I think, in Rio, at the Fiocruz Institute. So if you were there, or if you want to come, there are plenty of potential partners at that level. Also, there will be the announcement of the phase 2 results for the pediatric praziquantel, so worthwhile attending, I guess.
Who's the organizer in Rio? You know?
Eliana, from your point of view, from a partnership perspective, anything to say to potential partners?
Yes, mainly in the synthesis and the chemical synthesis. It's the most difficult one. We are working on this project now. Starting from the compounds we have identified, we have to synthesize to do to get more information, and this is the most difficult. But we are open to collaborations. We think to maintain the life cycle of schistosoma is not easy. We have very experience. We have 2 technicians only for maintain the cycle to the whole time, and this is not easy and not cheap also.
Brilliant. I think there's several questions. There's just one there with Professor Mark Gardner.
Hi, Mark Gardner from Salvensis. Thomas, I was wondering the very interesting PK/PD relationship that you found for praziquantel in the mouse model. If praziquantel was a compound coming to you through discovery now, and you had found this PK/PD relationship, would that speed up or slow down progression to the clinic, do you think?
If you would have known upfront about the PK/PD relationship, you mean? I would say the current criteria for praziquantel would have probably discarded the compound rather early. Knowing the PK/PD now, and given I would say the TCPs we would like to see, I still think that little chances that we will have progressed that compound. It's even though from I would say potent-wise, I mean praziquantel is not as potent as other compounds, so there are several parameters that would have probably reduced the progression. Of course, if it's the only choice you have, you push it, but knowing upfront, maybe not so much, right? So.
Now? Okay. I just wanted to follow on the comment how to take it further, and I'm not a schisto expert or anything, but I thought there's now a human challenge model, if I'm not mistaken, actually in Leiden. They just built a whole center about the challenge model, so I know you're in early stages, but perhaps that's a partnership. You may want to look into it.
Yeah, right, we're aware of that model. So just to complement on that, it's a model that uses only single-sexed schistosomes, right? So the caveat is that you need to make sure that your compound is active on two sexes, more in an equipotent manner.
There's a question just at the top. You can use the mic as well easier.
This is a question for Eliana. Do you know the target of your compounds, or the targets?
No. But to find the target is, I think it's the most difficult thing, because you have to do studies by binding your compound with some marker and to follow in the parasite. There are techniques using mass spectrometry to find the proteins that maybe the compounds are attached. We can, but so far the focus is to find, as we don't know the target, we are trying to find the structure. Because if you have the structure, all the functional groups that are needed are responsible for the activity, you have half the answer, and then you can find the target if you have the structure, the chemical structure first. And we are studying, now we are working on this, with in silico models to predict the activity. When we have a set of compounds, synthesized analogs, we will do the studies, structure relationship studies, and use these informations to design the better compounds, more active.
Yes. All the common downsides of natural product starting points. Yes.
Oh.
Thank you.
Time for a couple more questions from the floor.
I have maybe one for Eliana. Using seaweeds, is that coming from ethnopharmacology? Is there any ethnopharmacological background for using seaweeds as a potential source?
They use seaweeds for many, they don't use, there are many studies that identified some different kinds of biological activities, since antibacterial, antifungical, antitumoral, and because the metabolism of seaweeds is quite different from plants. They produce some kinds of metabolites with halogens, fluor, bromine. So they are interesting and different from plants. But there is no a drug developed from seaweeds, but they have a big potential technological, I think.
Is there any more questions or any more questions from the floor? Okay, so I think we'll conclude that panel. Thank you very much.
These worms. So brief background, but I don't need to tell the people in the audience here much about this, but just to say that there are a large number of people at risk of these infections, which are onchocerciasis and lymphatic filariasis, caused by Onchocerca volvulus, onchocerciasis, and Bushira bancrofti and Labruscia species for lymphatic filariasis, and severely disabling diseases. And the problems that we have with these, although there's been some great strides in terms of elimination for these diseases, there are still some hurdles that are difficult to overcome to reach those elimination goals, and that is mainly because current drugs that are used for the control programs only target the larval stages, the microfilariae of the parasites, that are largely ineffective against the adult worms. There are safety constraints, as Ivan alluded to earlier, with regards to loa-loa, because this can actually cause fatal adverse events if people have high levels of loa-loa in their blood when they're given ivermectin. And there's also emerging resistance to existing drugs. So a bit about Wolbachia and why we're so interested in it. Well, basically we know from a lot of studies that Wolbachia, these are these endosymbiotic bacteria. They're found, this is a cross-section of a female onchocerca worm. You can see these red stained bacteria in the hypodermal cords of the worm. They're also found within the developing embryos. So we know that they're needed for embryogenesis and larval development. We also know that they contribute to the adult worm longevity. This is a major barrier, the length of time that these worms live for. This is why the drug programs at the moment have to give the MDA for such an extended period, because these adult worms live for a long time. We also do know that the Wolbachia actually contribute to disease pathogenesis both through severe adverse events when worms are killed, the microfilariae are killed and release inflammatory components, and also clinical disease. So if you give 4 weeks of doxycycline, a broad-spectrum antibiotic, you can sterilise the adult worm, so they cannot produce microfilariae because of this problem with this need for Wolbachia for embryogenesis, and also blocks transmission, because any Wolbachia, any microfilariae that are released that don't have Wolbachia aren't able to undergo development in the mosquito. Then if you treat with this doxycycline, adult worms actually can die 1 to 2 years after treatment. This is a slow death once they have lost their endosymbionts, they're not getting all the things that they need from them, and they die quite a protracted death, which is actually a good, delivers a good safety profile. So it's macrophlericidal. And we also know that doxycycline improves pathology, both all through skin disease, bronchitis, also hydrocele lymphedema for LF. An important point is that this kind of treatment is safe in Loa endemic areas because Loa does not have Wolbachia bacteria, so it will be unaffected by this treatment. But why can't we be out there now using it? And it's because of a long course of treatment, so it takes 4 to 6 weeks. And also, doxycycline is contraindicated in pregnancy in children under 9. So AWOL, the consortium, the Anti-Wolbachia Consortium, was formed in 2007 to address these issues. And obviously 10 years is a long time in research, so I'm not going to have the chance to go through everything with you today. I just wanted to give you the highlights, to sort of give you the message of why we feel this is important, and also to highlight some of the new things that we're doing off the back of AWOL. So we have a screening funnel for drug discovery and development. We have these go and no-go criteria. Our screening starts with the cell-based screen, which I'll just highlight in the next slide to show you exactly how we do that. This has evolved over time. We originally had a 9-day assay with quantitative PCR and a 96-well plate, but now we can use 384-well plates and have a 7-day assay. We then move into in vivo models, and we're looking for a larval mouse model, which is a 14-day readout, and this is really trying to target and get a quick readout to see whether something is worth progressing into the more protracted adult Brucia screen in the mouse. So we're targeting the L3 to L4 stage, so seeing whether they're able to develop, and you get a quick readout from that. And we are always looking for greater than 90% Wolbachia reduction, because we have evidence from the trials that have been done with doxycycline that if you reach that threshold, you're leading to a macrophlericidal activity. And all of this is enforced by PK sampling and PK-PD modelling throughout, so that we're not progressing things that you have to give a really high dose in a mouse, and then we don't understand how that translates to the human situation. And then ultimately, what we're looking for in trials is a 75% reduction in treatment time course, so moving from 4 weeks to 1 week. And this is all aligned with our target product profile for an anti-Wolbachia macrophloricide that has 7 days or less treatment with oral delivery. And I just wanted to highlight, because I'll mention it now in terms of our screens, that we actually refined this workflow from what we did originally. Originally we had an in vitro worm assay here, and we took it out because basically everything that was in here translated into the larval model. Then we started screening more and more drugs and realised actually we need another way to triage the hits. So we had lots of hits, we needed to triage them, but also it was a way to confirm worm penetration. So if we moved something into the larval model and it didn't work, we then would wonder, is it because it didn't work, or is it because it just didn't get across the cuticle of the worm? So we're confirming that. We also confirm that it's a target in the worm Wolbachia, because their initial screen is done in an insect-based system. And we're also now able to do some other analyses, which I'll move on to speak about later. So just briefly, what our screening is we have a cell line that has Wolbachia in it. It's a mosquito cell line with mosquito Wolbachia. And we have an image-based operator system. We can screen thousands and thousands of compounds this way. But as I said, there's about 3 or 4 years worth of AWOL was developing this assay from our original screen, which was qPCR. So we're now in a much higher throughput place now. But we also reintroduced this microflare scheme, which is much lower throughput, so that's about 20 compounds a week, theoretically we can do. But there's a similar system where we can actually get EC50 analysis to find out how effective the hits that we find against the cells, the most promising hits we find against the cells, are in terms of their effects on the Wolbachia of microfilariae. And overall, we've screened against the cells over 2 million compounds, and we have over 20,000 hits. We also have our in vivo efficacy models, and basically what we do is, this is the larval strain, so this is the quick readout, which is 2 weeks. And what we're looking for, as you see with doxycycline, is you see this reduction in Wolbachia surface protein copy number. So this is a qPCR readout. So that just shows you that the Wolbachia has been reduced. You also see an effect on L4 length because they're not able to develop fully without the Wolbachia. Then moving into the adult model is a lot longer time frame, which is why we always filter through this first. But it's easier to get a readout of this, so things that come through here don't necessarily make it to being good hits at the end of this. But what we're looking for here is a reduction again in WSP, and you can also see the reduction in MF because of the hit on embryogenesis. So the first thing we did with AWOL was to test all the registered drugs. The human pharmacopoeia, which actually is surprisingly little, it's only around 2,600 drugs. Back then in the 2007-2008 when we started, we did this in combination with a company called Combinatorics. We then were also doing some small-scale anti-infective library screening from all of these different partners. I just wanted to highlight really what we've got, the most important things that we got out of this early screening, which was when we did the combinatoric screen, the human pharmacopoeia, the sort of usual suspects came out that we knew about, so tetracyclines and rifamycins, which we already knew were effective against Wolbachia. The fluoroquinolones had been tested previously. There had been some discrepancies between the different systems that had been published before, but they came out as being good hits against Wolbachia. Then from Abbott, who then became AbbVie, we got the Tylosin product that that Ivan from DNDi spoke about earlier. So what we did next was to take those tetracyclines, rifamycins and fluoroquinolones and move them into the drug development program of AWOL, whereas the hits entered hit-lead and lead optimization in the discovery program. So just to give you some highlights about rifampicin. So we knew, as I said, that rifampicin had good potency in vitro and in vivo models, but this didn't translate to clinical trials, because there had been clinical trials done with rifampicin and it hadn't resulted in the expected result. And we tried to establish whether this was down to a human-mouse mismatch, and we heard a bit about that problem with schisto as well. And actually it was found that through PK and PD modelling that in humans there is lower exposure than there are in mouse, so you don't get exposed to as much of the drug. And we've done some further work to show that if you use the clinically standard rifampicin dose, which was primarily defined in the past by the fact that rifampicin was only semi-synthetic and so it was very expensive. So the actual dose used was based on that, and you only get a 58% reduction in Wolbachia. However, if you do the high dose of rifampicin, then you get a 96% reduction in Wolbachia. Furthermore, we've also done some combinations of antibiotics with albendazole, which is one of the standard antifmalarial drugs, and found that if you combine rifampicin with albendazole, then it can actually produce synergy. So if I just highlight here the difference between using the standard dose, which is the 5, and the mouse is 5 mg per kg, and it doesn't get over this 90% threshold, but if you add the albendazole into the mix, then some of the worms are crossing over onto this 90% threshold. So we've managed to define that high dose rifampicin and rifampicin plus albendazole meet our TPP of 7 days oral treatment. Going back to the tylosin analogues, we went through some lead optimization of that. So as we found out earlier that tylosin itself, although a veterinary antibiotic, has very low bioavailability. So when you give it IP, you get this great reduction in Wolbachia. However, if you give it orally, you don't get the reduction. So initially we were thinking of changing the formulation, but then they got a chemist out of retirement who fancied about making some analogues of this, and this was very successful. We tested around 150 of them, and we found 2 analogues with improved potency and PK, which were selected for characterization. So you can see that they've crossed the threshold here at 7 days treatment. And this is now known as Tylomac with AbbVie, and as we heard earlier, this is actually now in safety trials in volunteers. So these analogues meet the TPP as well with 7 days oral treatment. In terms of other things that we've done, so those initial hits that I described there have come through the human pharmacopoeia and focus library screen, where it was all done on quite a small scale. They were anti-infective libraries, so we had some inclination that things would come out of that. And then we went on to using more diversity-based libraries to try and find things that we wouldn't have even have thought of testing in the first place. And we screened the 10,000 soft focus library from BioFocus. We've screened 500,000 compounds from the MMV library with medicines for malaria prevention. And we also have had a really successful collaboration with AstraZeneca, where we screened their complete portfolio of 1.3 million compounds. And this was done by basically seconding Rachel, who sat up there for a year to AstraZeneca, where we developed the screen further to be an industrialized scale screen to be able to screen all these compounds. And to highlight what has come out of this, first of all, Tylomac, as I've already described, the BioFocus library led to 6 series, so completely novel chemistry in the Antibobacter field, including template 1, which is AWZN1066, which we heard is now in the portfolio as macrofluoricides as well. We found 5 series from the MMV library. One of those was fusidic acid, which is another registered antibiotic that we didn't know worked against Wolbachia before. And we've done some work around this, but I don't have time to go into that in detail today. And then finally, from the 1.3 million compounds, we got lots and lots of hits from AstraZeneca, but we even managed to sort of titrate that down to 20 diverse structures with what is of interest now. And we're also still processing through the 68,000 global health chemical diversity library, which we have 185 hits from. So just to talk a bit more about these aspects of the hits that we have and how we're going forward in terms of finding out a bit more about them. So I described the microfluoride assay earlier, why we've reintroduced it as a means to triage the large number of hits that we had. But it's also proven very useful in terms of discriminating between very good hits before they go into the in vivo models. So our normal assay is using these Brucia microfilari. We treat them on day 0, and then we remove the microfilari at day 6, extract DNA, do qPCR. So basically the result we get with doxycycline and any good hits is that they reach an 80% reduction in Wolbachia. We then have introduced this new system where we're looking at the dynamics of this kill of the Wolbachia, where we wash off the drug at day 2, and we have also done shorter time frames as well, and then remove the drug at day 6, remove the microfilari at day 6. what we found, which is really interesting, is that all these nicely bright coloured ones on the left-hand side are the AstraZeneca clusters. And you can see in the dotted line here, it's showing you the 80% reduction that we would normally see at day 6. When you wash off the drug at day 2, you are still able to achieve this 80% reduction. And this is the case also for template 1, which is our 1066 compound. Not quite the case for Tylomac, and also all the known antibiotics that we know work against Wolbachia aren't able to achieve this 80% reduction with only 2 days of treatment. We have actually gone less, and it looks like it's somewhere sort of 12 hours plus that we can start to see some really good activity with these compounds. So we have 5 AstraZeneca clusters and AWZ-1066 that have accelerated kill profiles, which suggests a different mode of action. So the key to this is that when you're only looking at day 6, they all perform exactly the same. So all of these will be down here at day 6, but if you wash the drug off at day 2, only these AstraZeneca compounds in 1066 have this accelerated reduction in Wolbachia. So we're interested in researching this a bit more to see what's going on, because they have a different mode of action. And aligned with that, we also are doing some target identification work. So you were being asked, Eliana, earlier on about, do you know the targets, and this is where we're going to now, because actually you don't need to know the target when you're doing these phenotypic screens, just trying to find things that work. But now we're at a point where things are going through regulatory approval, and these are the kind of questions you get asked, of what do they actually target? So unfortunately, Wolbachia is genetically intractable, which makes it difficult to actually do sort of genetic target identification processes. So we are currently using a chemical proteomics approach with photoreactive probes that we've made to 1066 and Tylomac, and we're hoping to take this forward with our other hits that we have. But just to highlight the process, so basically what you do is you incubate cells, and we're also extending this work with the worms, where you incubate with your probe, which looks very much like your drug, but has like this click handle on it. And you incubate, you compare that to DMSO, you use UV light to create a covalent bond with your probe and your target proteins. And then you're able to do pull downs because you attach biotin, and you can pull it down with beads. And also you can look at in-gel fluorescence to see what binding, and also perform mass spec to identify the proteins that have been pulled down in this approach. And what we're just an example of some of our results, where you have proteins that are enriched with the probe but not with the DMSO. So we're following this up just now in collaboration with Imperial College London and AMPI, and the chemists at the University of Liverpool who are making the probes. So just to summarise at the end, so when we started this consortium in 2007, we had only one drug, which was a proof of concept drug, and now we have this portfolio where we have doxycycline is now being used in particular areas in terms of policy, and we have some other proof of concept drugs, and we now have all these candidates that we're hoping to take forward. And Tylomac is, as I said, in the DNDI portfolio in phase 1 trials. We also have all of these things to back up, but like we've been talking about earlier, the funding for this part is not really there at the moment, so that's the difficulty if there's attrition down the pipeline. And there is a cast of thousands, but again, just to highlight the number of people that it takes to form such a consortium to get through to that final slide in terms of portfolio.